I am Dr. P. Uma Maheshwari, Head and Associate Professor, Department of Botany, Sri Sharda College for Women, Autonomous, Salem 16. In this video, let us see about mutation. In this video, we are going to discuss about introduction, history, characteristics of mutation, classification of mutation, and types of mutation with questions. And let us see the introduction of mutation. Mutation is a heritable change in genetic material or character of an organism. Showing these changes are known as mutants. That is, the individuals with changes or modifications are called as mutants. The individual showing an altered phenotype due to mutation are known as variant. The agents which causes the mutation are called mutagens or mutagenic agents. Mutation which causes changes in base sequence of a gene are known as gene mutation or point mutation. The history of mutation. Seth Wright was the first person, he is an English farmer, recorded the case of mutation first time in the 1791 in male lamb with unusual short legs. And the term mutation is coined by Hugo Duveris in 1900 by his observation in Enothera. The systematic study of mutation was started in 1910 when Morgan genetically analyzed white eye mutant of Drosophila. Then H.J. Muller induced mutation in Drosophila by using X-rays in 1927. He was awarded with Nobel Prize in 1946 for his work. What are the characteristics of mutation? Let us see one by one. Generally, mutant alleles are recessive to their wild type or normal alleles. Most mutations have harmful effect, but some mutations are beneficial. The spontaneous mutations occurs at very low rate. Some genes shows high rate of mutation. Such genes are called as mutable gene. Highly mutable sites within a gene are known as hotspots. Mutation can occur in any tissue or cell, either in somatic or germinal cells of an organism. Classification of mutation. Based on the survival of an individual, the mutations are of 5 types. First one, lethal mutation. When mutation causes death of all individuals undergoing mutation are known as lethal. Sublethal mutation causes death of 90% of the individuals. Subvital mutation. Such mutation kills less than 90% individuals. Vital mutation. When mutation don't affect the survival of an individual are known as vital mutation. Supervital mutation. This kind of mutation enhances the survival of an individual. Based on the causes of mutation, there are 2 types. First one is spontaneous mutation. It occurs naturally without any cause. The rate of spontaneous mutation is very slow. Example, methylation followed by deamination of cytosine. Rate of spontaneous mutation is higher in eukaryotes than prokaryotes. Example, UV light of sunlight causing mutation in bacteria. Next one is induced mutation. Mutations produced due to treatment with either a chemical or physical agent are called induced mutation. The agents capable of inducing such mutations are known as mutagen or mutagenic agent. Use of induced mutation for crop improvement program is known as mutation breeding. Example, X-rays causing mutation in cells. Based on tissue of origin. Two types, somatic mutation and germinal mutation. The mutation occurring in somatic cell is called as somatic mutation. That is, the vegetative cells or the non-reproductive cells. In asexually reproducing species, somatic mutations transmits from one progeny to the next progeny. Germinal mutation occur in gametic cells or reproductive cells are known as germinal mutation. The sexually reproductive species only germinal mutation are transmitted to the next generation. Based on direction of mutation, forward mutation and reverse mutation. When mutation occurs from the normal, that is wild type allele to mutant allele, are known as forward mutation. Reverse mutation, when mutation occurs in reverse direction, that is from mutant allele to the normal or wild type of allele, are known as reverse mutation. Type of trait affected: visible mutation and biochemical mutation. Visible mutation, which affects on phenotypic character and can be detected by normal observation, are known as visible mutation. The mutation which affect the production of biochemicals and which does not show any phenotypic character are known as biochemical mutation. Let us see one by one the types of mutation. Chromosomal mutations. It involves changing the structure of the chromosome or loss or gain of the segment of chromosomes. There are 5 types. One is deletion, inversion, translocation due to non-disjunction and duplication. Let us see one by one. First one is deletion. It's due to breakage of the chromosome and a piece of a chromosome is lost. Here is the example. In this portion, this is the original chromosome. Here there is a break or loss of a segment of chromosomes. So this portion gets deleted and the final product of the chromosome is like this. So this is called deletion. That is a loss of a segment of a chromosome. Yeah, that is a piece of chromosome is called deletion. Next one is inversion. It is the chromosome segment breaks off and flips around backwards and segments reattaches. So here there is a breaking off or break off the chromosome point. That this pass gets inverted and reattached with the original chromosome. So here the genetic material gets reversed and the genes are arranged in inversion manner. Next one, duplication. It occurs when a gene sequence is repeated. Here the chromosome structure, this portion gets duplicated and the portion increased in this chromosome. A section of DNA is duplicated and the length of the chromosome gets increased and the genes are also get duplicated. Next comes translocation. It involves 2 chromosomes that are not homologous chromosomes. The non-homologous chromosomes gets translocated. Part of one chromosome is transferred to another chromosome. The non-disjunction, that is, failure of chromosomes to separate during meiosis, and it causes gametes to have too many or too few chromosomes. Let's see the non-disjunction process. This is the normal meiosis structure. It starts normally, tetrads formation and line up in the middle. And the next process, one set of chromosomes moving towards the pole. And during that time, here the separation is not equal. The one set of chromosomes in this is going to the upper pole, and this red color means that one set of chromosomes goes towards the lower pole, whereas in another set of chromosomes both the chromosomes are moving towards the upper pole. So after this process, the meiosis 2 occurs normally, and all gametes have an abnormal number of chromosomes, either one too many or one too few. Here we can see this one again under second meiotic division, it's going like this. So this set of chromosomes enter into one gamete and only one chromosomes enter into this gamete. So during that time, this set of chromosomes enter into one gamete becomes n plus 1, and like this n plus this gametes formed from this gamete, that is after the meiosis we'll get 4 gametes or 4 sperms or 4 things. So in 4 daughter cells, so 2 of the 4 daughter cells are having n plus 1 chromosome, where the another 2 daughter cells are having n minus 1 chromosomes. So due to non-disjunction also the process is taking place. So here we can see the animation of the chromosomal mutation one by one. The original chromosome and deletion, a part of portion gets deleted. Inversion, here the arrangement is changed into another order. Translocation, it got joined with another non-homologous chromosomes. So here you can see the original chromosome structure, or original chromosome is having the gene order like this. We'll assume this A, B, C, D, E, F, G as a gene. So this is the original chromosome. Here the first process, duplication, is taking place. So the gene D gets duplicated and in 2 or double dosage. So addition of one particular gene is added to the chromosome. The second one, deletion. Here the gene D is not in the chromosome. So it gets either due to break off or some other causes, it's not in the chromosome, deleted from this chromosome. Then inversion process. Here this E and F gene gets inverted over that. So the order of E and F is get changed. Then inversion. Here A, B, C is the order, but instead of B and C gets inverted in the chromosome. So I'll sum up the gene mutation. It's a change in the nucleotide sequence, and it may involve a single nucleotide and may be due to copying errors in chemicals. Types of gene mutations. First one, point mutation, substitution mutation, insertion, deletion, and frame mutation. Point mutation. It's a change in a single nucleotide. It includes deletion, insertion, or substitution of one nucleotide in a gene. We have already seen the deletion and insertion. So the example for point mutation is a sickle cell disease, the result of one nucleotide substitution. It occurs in the hemoglobin. Next, frame shift mutation. Here, inserting or deleting one or more nucleotides and changes in the reading frame like a change in a sentence. We'll see the. So due to that, the proteins built incorrectly. So here, an example, frame shift, how the nucleotide gets changed. With this example, the original, it may be FAT, CAT, ATE, THE, WEE and RAT. After we added the A in between the nucleotides, that is we are assuming this as these are the nucleotides. So here we are including or inserting or adding the one gene A. So the CAT becomes CAA. Here we have added, so this T joined with another word or another nucleotide, that is triplets, AUG or CCG, like that it will be there. So it added to the another triplet, so it becomes TET and HEW and EER and finally only with the two letters. So during the frame shift mutation, the nucleotide gets arrangement changed. So here is the example, amino acid sequence gets changed due to the frame shift mutation. So this is the original arrangement of the nucleotides. The ATG will give methionine, GAA will give glutenine, GCA alanine, CGT glycine. So here there is a change in this. So this will be like this, ATG methionine, it will produce the amino acid methionine. Instead of G, here there is a shift of this nucleotide. G has gone. So this A added with G. So it becomes AAG. It will produce lysine. Instead of giving gluten, it is producing lysine. Another combination is CAC. Here CAC. So instead of producing alanine, this will produce histine. So here this will go like this. It will combine with the another nucleotide. So this is the way the frame shift mutation is taking place, through which the production of amino acids gets changed and it will lead to the protein production also. So here animated video with this process. So this is the original order. Substitution is taking place. Then insertion of this, deletion of that process. So like this the nucleotides arrangement gets changed. So this video we have seen the introduction of mutation, history, characteristics of mutation and classification and types of mutation. Over with, by discussing this question, we'll clearly get an idea about the mutation. So by answering these questions, that is define what is mutation, define mutation and explain its types. What are the types we have studied, and gene mutation, how it brings the changes in the organism, and how we can differentiate the deletion from insertion, translocation process, and in the chemical mutations the tautomerization process will come. The gene sequences changing frame shift mutation, the base analogs production of amino acids through which the protein synthesis also gets changed. So by analyzing and answering and discussing these questions, we'll get a clear idea about the mutation. Here I have given the link for study material and questions for your further study. And thank you all for watching this video and listening this video. Hope you will get an idea of mutation. Thank you.